Oh, welcome everybody. I'm really excited to welcome you to this afternoon, this morning's progress update or webinar. My name is Colin Pele. I'm a clinical pharmacologist. I'm talking to you right now from Basel, Switzerland, and I've had the pleasure of being part of a team that has put together this applied pharmacometrics training program. So if you could go to the next slide, Amira. So this is a webinar. So if you've joined as a delegate or as an attendee, by default, all attendees' videos are disabled and you are muted. So don't get worried when you see that. We will be monitoring the attendees so we can see as you come into the room. And when we go to the open Q&A section, if you need to talk, we'll be able to unmute some of you. Please use the Q&A feature if you have a serious question. At the moment, I can't see the Q&A section. Oh yes, it's there, it's available. So use Q&A in Zoom and use chat for social stuff. So feel free to just go wild and say how excited you are to be in this meeting using the chat. But if it's a serious question, then again, part of the team that's here will be monitoring that and will then present your questions during the discussion session. So I'm a big fan of these immersive learning experiences, and this is what the Applied Pharmacometrics training program is about. It's a short, focused program where there's some teaching, learning. There's some what I call healthy stress. The people in the room there, they're beginning to smile because I guess they say there's no such thing as healthy stress. But essentially, strict timelines is one of the key things that we have within this program to try and mimic what a typical drug discovery or development project would be about. But I'm not going to take the thunder away from Amira and Ochuko, who will be the first two speakers, and they will describe to you the program. I just want to say that I'm very excited that Rick de Greef, Craig Rayner, some of my colleagues, we met way long ago in a meeting in Seattle where we conceptualized actually this program. So I'm very excited that we're able to continue to run it for this period of time. After Ochuko and Amira, we will then go to the main topic here, which is to go through for about 20 minutes presentations from each of 4 teams that will present their projects to you. So with that, oh yes, I want to say one more thing. Social media, we actively encourage that. So if you see something that resonates with you and you want to pop that onto LinkedIn or make a post about it, feel free. The hashtags that you could use is hashtag APT or hashtag PMXAfrica or others that you might find relevant, like clinical pharmacology, pharmacometrics, etc. With that, let's go to the next slide, which is probably the agenda slide. So here's the plan. We will try as best as possible to keep to this time. So I still have 2 minutes of talking time, but I'll probably give that to Ochuko and Amira to get started. And then we'll do 2 presentations before a coffee break, and then another 2 presentations, and then an open discussion, and we will close on or before 6 o'clock Central European Summer Time or South African Standard Time. With that, on to the next slide. Over to you, Ochuko.
Thank you, Colin. Yeah, so as a proud nurse of the APT, I will be taking you through what we do here and what our outputs have been over the years. And when I say over the years, I mean from when we conceptualized this, from when this started, the first cohort in 2022, and now we're in the 4th iteration, 2025. So the APT program is a graduate level fellowship. Open to MSc and PhD students, and of course postdoctoral scientists, but must be from of African origin. It's a hybrid program that operates both virtual and on the application of research and development and clinical applications. How do pharmacometrics apply? How do we bring it into our clinical setting? And of course, streamlined to what operates in Africa, what is applicable in Africa. Over the years, the program has evolved. And right now, what we have in 2025 is that we had the fellows first go through an intensive and a very intensive theory section, which then prepared them for what they did in their projects. And I tell you, when I said intensive, it was really intensive and rigorous. I can tell that from my hand. Even the faculty can tell how rigorous it was for both the fellows and the faculty. So apart from just learning about pharmacometrics, there's also the instilling of leadership skills and communication skills. That comes very much alive in our physical meetings, which we call face-to-face. That is how we target face-to-face meetings, where it's still because we have to communicate our work to the larger audience. So there is the communication skills that is imbibed. Leadership skills come very much alive in this meetings. And of course, you can't be in a room with Colin and not have the leadership skills seep into you unconsciously. It's a well-rounded program that we go through for 6 months. For this one, we first had a phase 1, a pre-selection phase, a phase 2, the theory, and the phase 3. And this we ended up with 10 fellows across Africa, which you'll be listening to what they did in the phase 3, which was the project phase. For a major part of the work of a major goal of the APT program is that the outputs of this program culminates into a publishable unit. Next slide, please, which you will see. Over the years, we have had our fellows publish our outputs, and we are very focused on high impact journals. Whatever we are doing on the APT program must be worthy of publishing in high impact journal, peer reviewed. From the 2022 cohort to up to this cohort, we've had our fellows publish their work. For instance, you see from the of your screen, the bottom left side, that's a project that was done by a team member, a team from the 2022 cohort. And for my team, I am a 2023 alumnus. We took the work further in the 2023 cohort, and we published what we have in the middle top paper, what you see there. And even for the 2024 cohort, they've also started publishing their work. That's one of those is what you see in the bottom right column of your screen. But much more than publishing our work, it's also that we showcase our publications, our outputs in international meetings. Please, the next slide. We showcase what we do in international meetings, conferences, not just because we are not local. We do our work to international standards, and so we are able to attend these meetings. Thankful to our sponsors who make this possible, that offer travel grants to our fellows to attend this international meeting. From 2022, from WCOP 2022 to page 2023 to page 2024, and even to Pharmacokinetics UK, the Digital Health Africa 2024, and to the most recent, the inaugural Pharmacometrics Africa conference that took place in September in Kampala just this year. All this platform exposes the fellows to international collaborations that have actually moved our fellows even in their career. People have gotten international collaborations that have moved them forward in their careers, which you will see in the next slide. All this, we are able to present our work. Poster oral presentations have come out of the APT program of high standard. Right now, I will now hand over to Amira, who will take us further in this discussion. Thank you.
Thank you so much, Ochuko. My name is Amira Ghunaim, and I'm an alumni from the first cohort 2022. As Ochuko has mentioned, the program Real Power lies in its impact on the ground. So this program strengthens the skills of the local African scientists, and the fellowship helps retaining the talent in Africa through addressing existing training gaps and fostering job creation. By saying that, if we look at the map on the left, this shows the distribution of the fellows and where they're from. And you can see that they come from starting from North Africa all the way to South Africa. I can say that the majority are from Nigeria, Kenya, Uganda, Egypt, and South Africa. And also it's noteworthy to mention that the majority of the fellows come from academic backgrounds. But usually during conferences, we're asked, where are the fellows now? So on the right, it's just a small description of where the alumni are working. Some of them had postdoc and research positions in universities such as Stellenbosch, UCSF, Gulu, and Rhodes University. Others wanted to join the industry and the pharma, and they were able to get positions in Novartis and Eli-Lilly, whether in quantitative sciences or in regulatory affairs. Also, 3 fellows two work in Certara in drug development solution, and others in Pumas AI. And the most important thing is that at the end, the APT fellows contribute to their wider scientific communities in different ways. It could be through mentoring the new cohorts or helping in organization of conferences and webinars. And thank you very much. With that, we'll go back to Colin.
Awesome, that's great. Thank you. And Amira, as you were saying that, I'm looking at the attendees, and several of your colleagues are actually in the room. So it's really nice. And thanks, Rick, for the comment. Just to repeat something that I said earlier during housekeeping, since this is a webinar, most of you would have your videos and your audios automatically disabled. So please use the chat to say what you feel socially, and then if you have a question, put it into the Q&A. With that, let me hand over to Innocent, who will introduce the first 2 teams that he's been actually supporting as well. Innocent.
Thanks, Colin. As I'm Innocent Asimwe, a pharmacometrician at Novartis UK and an alumnus of the program. So welcome again to the webinar. Please join me to welcome Ibrahim Komeil, who's an assistant professor of pharmaceutics, and Nouran Nassar, assistant professor of clinical pharmacy, both from Egypt and representing Team Eli-Lilly 1. So in collaboration with Eli-Lilly, their project focused on modeling antibody drug conjugates, that's ADCs for oncology. ADCs combine antibodies with chemotherapy agents to precisely target tumor cells. The team applied quantitative systems pharmacology, QSP models, and user-friendly analytical tools to evaluate dosing strategies and support the optimization of future ADC therapies. I'll now hand it over to Ibrahim and Nouran.
Okay, can everyone hear me? I hope the slides are showing. Okay. Good evening or good morning, everyone. It's a pleasure to be finally presenting our project for the APT Fellowship for 2025, which is titled Modeling Antibody Drug Conjugates in Oncology. This presentation and project has been prepared and will be presented by myself, Nouran Omar, and my teammates, Ibrahim Abdel Mohsen, under the mentorship of Venkatesh and Daniel from Eli-Lilly, as well as Innocent from the APT core program. So we will start with presenting an overview of the scope and the objectives of the project, and then I will hand over to Ibrahim to continue describing the workflow of the project. I'll start with an overview of the importance of antibody drug conjugates in the context of chemotherapy. So ADCs, they have dual benefit of targeting specific cancer cells according to the corresponding receptor, as well as the efficacy or the potential to undergo cell killing for these cancer cells due to the cytotoxic portion of the ADCs. This selective targeting of those tumor cells not only enhances the efficacy in chemotherapy, but also helps reducing adverse effects as well as systemic toxicity. And this means that it's potentially valuable for treating metastatic and refractory cancers. It is due to these benefits that there are multiple approved ADCs, and there's also a very active pipeline under development as an attempt to improve precision delivery of chemotherapeutic agents to cancer cells. So despite the benefits of ADCs, they do have their complexities, and this complexity arises from its tripartite nature. So they consist of 3 different components, and each individual component has its own pharmacokinetic and biotransformation properties. So the first component is the antibody, and this is responsible for the selectivity of this ADC, where it binds to the corresponding antigen or receptor that is highly expressed in the cancer cell and is less expressed in non-cancerous cells, thus providing the specificity and selectivity in the target behavior of the ADC. The second portion or component is the linker, and this links the antibody to the payload. This linker can either be cleavable or non-cleavable and is responsible for the eventual release of the payload. And the final component, which is the payload or the warhead, and this is responsible for the actual cell killing. It has the cytotoxic effect, and it binds to the target site inside the cancer cells, whether it is the tubulin or the DNA, to result in cell killing. The number of payloads that are attached to each antibody is identified by a very important key parameter, which is the DAR, or the drug-to-antibody ratio. So it's important to understand each of these individual components to be able to optimize them in order to enhance the efficacy and the safety of ADCs for drug development. In order to address this complexity, there's a suggestion that we use mechanistic modeling at the pharmacokinetic cellular level in order to be able to describe these processes and behaviors of these species. This approach is called quantitative system pharmacology, or QSP. So this mechanistic level modeling, it uses different differential equations to walk us through the different processes and behaviors. It starts with the antibody portion of the ADC binding to the specific receptor that is on the surface cell of the cancer cell. And then it is internalized through the endosomal internalization and then lysosomal degradation in order to release the payload. And finally, this payload is attached, or it binds to the target cell killing portion of the cell, whether it's the microtubule or the DNA, resulting in cell killing. So to understand these behaviors, it's important in order to enhance dose optimization, to be able to understand the payload distribution inside the cancer cell at the granular cellular level. So let's look at what the literature is focused on. The current literature is mainly focusing on ADCs that target the HER2 receptors that include ADCs such as trastuzumab. And unfortunately, there are limited PK cellular models that include other ADC targets, such as the emerging folate receptor alpha target and the Nectin-4. So the folate receptor alpha is very important because it's expressed in solid and uterine tumors, and it has mirvetuximab as an approved ADC, as well as the Nectin-4, which is also expressed in urothelial breast cancer with enfortumab approved. Those 2 emerging ADC targets are, however, lacking in terms of comprehensive cellular PK models. But this helps us with a research opportunity where we can actually develop a mechanistic cellular PK model that can predict the behaviors of those target ADCs that are not just the HER2 receptor ADCs, and improve their drug development. That brings us, of course, to the project objectives. So the first objective is to be able to implement literature-based models or PK, pharmacokinetic cellular models for those ADCs. Also, to extract cellular PK level pharmacodynamic data for ADCs in order to be able to validate these selected models for accuracy. Also, develop interactive tools such as R-Shiny applications as an open source option for allowing visualization of parameters and providing also an interface for those with limited coding experience. And most importantly, to select the model that can best predict the pharmacokinetic parameters of ADCs that are that have targets other than the HER2 receptors. So in order to be able to achieve these objectives, I'll start walking through the workflow for the model implementation. So we're using a stepwise approach where our first step is compilation of those models from the literature, where we extracted the ODEs for these models, as well as system parameters, drug-specific parameters, experimental initial conditions, and of course, along with their units and physiological ranges. After extracting these ODCs, we'll be implementing these ODEs in the second step in translating it into molecular syntax to implement it in monolix application. And then this is just to confirm that we are confident with our replication of these models and comparing it with the published reported data. And then the next step will be translating in R syntax for RxODE simulation and implementing these ODEs again. And finally, using or developing an R-Shiny application in order to allow real-time parameter exploration and simulation. So I'll hand it over to my colleague Ibrahim, who will be walking us through the literature-based PK cellular models and will provide a comparison. On to you, Ibrahim.
Thank you, Nuran, so much for your presentation. Hello, everybody. Good afternoon for everyone. So as Nuran mentioned, first step is to extract the ODE from the literature in order to use it in building up our cellular PK model. So we have here in the shown slide the list of summarize of the 4 important models that we gathered from the literature. We started with the first one, Shah et al. He published this article published 4 differential equations explaining the ADC cellular PK profile targeting CD30 receptors, representing a simple mechanistic kinetics. However, there is no endosomal modeling mentioned in that article. The second one was a Singh et al. Article. It's published 8 differential equations explaining bystander effect for EDC targeting HER2 receptor. But what is the meaning of bystander effect? The bystander effect means that if a small payload released from the ADC can not only target that particular cell, but they can also diffuse to adjacent cell, exerting their effect on antigen-negative cancer cells. Thus, there is a complex parameterization is noted in that case. The third one was published by Matt et al. Demonstrating 6 differential equations for also HER2 receptors, but intracellular payload trafficking was limited in that study. Finally, there is a Schuller et al. Article which published 13 differential equations targeting HER2 receptors, also explaining different processes related to EDC cellular kinetics, except that the DNA payload binding or cross-linking was not explained in detail in that study. For this, we decided to use Shah et al. As our starting point because it provides a solid basic framework. After that, we plan to build on it and move to Schuller model, which is more complex and comprehensive compared to Shah model. Next. So, as I mentioned, for Shah model, it is simply explains the cellular PK profile in 4 different compartments, as extracellular representing the amount of the EDC in the extracellular compartment, cell surface bound representing cell surface binding receptor with ADC payload amount either intracellular or extracellular. Next. After understanding the model serial kinetics, the initial estimate should be identified as shown in the table, where K on and K off ADC represents ADC receptor binding and dissociation rate constant. K out PL represents the payload efflux rate constant out of the cell, total surface antigen concentration, internalization rate of the receptor Kint, and drug antibody ratio DAR, and finally the total cell count used in that experiment. Next. By using WebPlot digitizer, we extract the dataset through digitizing the intracellular, which is shown in that figure on the left, and the extracellular payload, which is shown on the right. For the Oakley et al. Paper, it is the same paper used in that in the modeling in the Shah modeling, focusing on Carpus cell line 299, targeting a CD30 receptor. Next. The fourth step is model building, which was taking place through two different softwares. First software was Monolix in order to check the validity of the ODEs as shown in the left-hand side and to replicate that model. And as shown in the right-hand side of the slide, we got a good output as shown for extracellular and intracellular payload. Now we are confident to move forward to use that ODEs in simulation using our software. Next. So by using our XOD package, we are now able to use the model ODE block to simulate the 4 output parameters like ADC extracellular and cell surface bound ADC, in addition to both payload extracellular and payload intracellular. And because we have only a data set for the extracellular and intracellular payload, we can obviously get the same outputs compared to published data by Shah model. Next. As shown in that figure, RxOD simulation showed in blue color is in agreement with the published simulation by Shah et al. Model, showed in orange color. And because it is a simplified model and there is a very limited equation describing the complexity of the cellular kinetics of the ADC, we can figure out here there's a deviation from the model data and the observed data. Thus, we need more equations to detail describe the cellular kinetics for the ATC using for the cell line. Next. After we are now confident about our model building, so then we moved forward to create a Chinese app to be used by non-modelers to simulate ADC PK easily in an interactive way, as it allows a real-time exploration for key parameters. Next. Based on that, we successfully simulated 4 outputs for Shah model using ODEs representing extracellular ADC concentration, cell surface bound ADC, intracellular and extracellular payload concentration. In addition, we translated that into an interactive R-Chinese application. So I will back over again to Nuran to complete the rest of presentation. Back to you, Nuran.
Okay, thank you, Ibrahim. So to wrap up, and as a key message from the outputs of this project is the CHABETS model was able to be successfully implemented and replicated with agreement, of course, with the published results or the published data, and also overlay with the data sets that were published in literature. Despite being a simple model with only 4 ADCs, however, they were able to capture at least the ADC cellular behavior or the key cellular behavior for ADCs and the trend in the pharmacokinetic cellular modeling, providing accessible open source tools such as R-Shiny applications can assist in parameter exploration and visualization. And it's an option for non-coders. And also it's important that this model or the CHABETS model or the simple model is a preliminary foundation model that can be used for non-HER2 ADCs, predicting the pharmacokinetic cellular patterns and behaviors for emerging therapies and ADC targets such as folate receptor alpha and Nectin-4, providing a tool for supporting ADC drug development. And this brings us to the next steps. So the next step will be expanding this model. We're building on from the SHA simple model into our more complex second candidate model, which is the Schauer-Betz model, which includes more ODEs and differential equations describing the ADC behavior, and also concluding it with developing an R-Shiny application for interactive exploration. And then we will compare this complex model with the simple SHA-Betz model and validate or use PK cellular data sets that are describing the pharmacokinetics of non-HER2 ADCs, such as folate receptor alpha and Nectin-4, and comparing the two models in order to select the best model that will help predict these published or these data sets for those non-HER2 ADCs. So thank you very much. This brings us to the end of this presentation. I'd also like to take the opportunity, of course, to thank everyone. I'd like to thank the APT team for this opportunity as being a cohort for the 2025 fellowship program. And I'd also like to thank our sponsors for their support, Venkatesh and Daniel, as well as the continued support of Innocent. I'd like to thank Colin and all the APT mentors, Samer, Amira, Ochuko, Vinand, and of course, my colleague Ibrahim and all the APT fellows. Thank you again. Thank you very much.
Thank you so much. Thank you.
Thank you, Ibrahim and Nuran. So we started a bit early, so we have some time for questions. Please, can you use the Q&A? So far there is a question in the chat. It's a comment, but again, more or less a question. So the Ray says that the model doesn't seem to fit the observed data very well. Any comments on that?
Yes, I will go to answer for that comment. Yes, we agree with you that there is not model fit very good, and that is because it is a simplified model. It is not detailed explaining the full ADC kinetics. As I mentioned in my presentation, it is a simplified model, and it is also only very limited equations. And as my colleague Nuran claimed, that on our future plan that we are going to build on that model in order to explain that ADC's full cellular PK kinetics using more detailed equations in order to catch up all the compartments that might be affect on the cellular kinetics for the ADC. So we agree with you, and thank you again for your comment.
Okay, thank you. Another question from Roland. So Roland from Stellenbosch. Thanks for the great presentation and work done. Do the ADCs that were considered induce anti-drug antibodies? If so, should it be incorporated in further work?
I'm sorry, what's the question? Can you repeat the question again, Innocent?
So the question is about anti-drug antibodies. Did the ADCs considered induce them? And if so, should this be incorporated in further work?
Okay. So would you like to go for it?
As you think about it, I've noticed your sponsors from Eli-Lilly.
Yeah, maybe I can chime in again.
Yes. Thank you.
Thank you, Venkatesh.
Yeah, so for ADA or immunogenicity risk for ADCs is very, very low. So based on approved ADCs, there is none of them had, other than I think Kadcycla maybe had a very low incidence of ADA. Other than that, Kadcycla ADC, which was approved maybe 2 decades ago, there's no signal of ADA formed, especially for the example which Nuran mentioned, the ADA incidence is very low, but it's a good point. If you see the difference in the kind of PK in the clinic, we could look into, but ADA in the in vitro system, maybe not relevant. But I think the ADA formation in the clinical settings could be helpful to look into. But for the in vitro, I don't think, because we are incubating only for 3 days, I don't think ADA will be formed unless they have some kind of a baseline ADA formed within that particular cell lines. I don't think there will be any impact.
Thank you for that. Another question, and how does the model impact TMDD on the disposition over time? So this is about target mediated drug disposition.
Yes, I may answer for that question too. Our focus, we are now focusing on the cellular PK modeling, and there is another models that we are going to planning to do after we already reach a good and representable model related for the cellular PK. We have to make this model to TMDD. But we need first that we can to use a very good model in order to explain the detailed kinetics for the ADC before we going to TMDD. But sure, it will be a need that we have to do that also.
Yes, I'd also like to add that we start always from the basics. So yes, we were focusing on PK cellular, and this is the foundation. And all the published models, usually their initial step is PK cellular, moving on to PK in vivo, and then PK/PD modeling, and finally into clinical translation. So I think we're moving in the correct stepwise approach. And at first, we have to start from the basic foundation. And as Ibrahim mentioned, we select the best model and then build onwards forwards.
Okay, thank you. And yeah, Samia has responded to that. So if you check the Q&A, there are more responses from Samia. And okay, so I'll give it a few more minutes if there are any more questions. As we wait, I'll ask you this. Could you share your perspective on implementing the QSP model in RO monoliths? Any key challenges or insights from a modeling standpoint? I know this was your first time to work with models, and then QSP, you are looking at, it's like at the extreme end. So any key challenges or insights?
Yes, and you mean like for the simple model or the future work?
For the
because yes, this was our yes, this is our first experience working with ADCs in general. For me specifically, I'm not used to using or working at this granular level because I'm a clinical researcher, and that's why it was really interesting to understand the ADC behavior and to understand it at this very particular accurate stepwise level. And there was an experience, or there was a challenge in replicating the published work. This is what I can discuss with you. And as we've noticed, simple models, they probably prove that they're less can sometimes be more. Even if they are simple, they can be easily replicated, and they're feasible, and they can be efficient. And complex models are, even though they're complex and they probably cover a lot of different processes for the ADCs, but however, they may not necessarily be replicated easily. And they not necessarily mean that it's a one-fit-all. It could work with a certain ADC, but does not necessarily mean that it can be replicated, or it can predict the behaviors of all other ADCs. So that was really challenging. And I think that what we learned from this experience is that we were able to model using different softwares, being able to replicate those really and translate those ODEs that are really difficult, and that from the start of the project, they were just equations that were very complex to understand, and then to be able to understand them and actually replicate it, I think that's a good achievement. Ibrahim, would you like to add something?
Yes, so in addition to Nuran's words, yes, QSP modeling is very challenging, especially for the ADC. It is very complex and it is not easy compounds, and it is very important nowadays in the drug development process. Yes, it was very challenging for us for replicating the model, and we found many of articles that like to have a certain point of view different from the another article, that can like to express in more details kinetics and also dynamics for the ADC either in vitro or in vivo. But the most interesting in the modeling itself, because it is now we can like to expect and predict the concentration for like the complex of the ADC with the receptors, and also concentration and also the amount of the payload intracellular or extracellular or either inside the endosome by use of modeling. Regarding that, there is no article that we gathered can measure everything in every compartment in the cell. So each article like to measure like specific compartments, and it's now by have a good representative model, it is now possible that we can like to use it, especially that we have already the Shiny app, so we can use it in order to predict the actually concentration of the different components of the ADC in different compartments across the cell. And it is also make sense for others models for the in vivo level too. So it was very challenging project, and thank you, Venkatesh, for your endless support, and also Daniel and our mentor Innocent from the APT for all of this work. It is nothing to happen without your kind support. So thank you again.
So thanks again, Ibrahim and Nuran. There is one last question. So compared to the published model and data, what was the sample size and richness of the PK data? So as you think about it, if you have a short answer, if not, you can please type the answer in the Q&A forum.
Okay.
Any quick answer?
So this was PK in vivo cellular data. So it was just based on in vivo experiments on a certain cell lines. That was the data. These were the data sets that we used during the project. And I'd like to also highlight that there is a gap in literature in terms of PK cellular data sets to use for ADCs other than the HER2 receptors. And I think this was also one of the challenges in our project.
Okay,
Ibrahim,
thanks.
would you like to add?
Thanks again.
Okay.
So yeah, before we move on to the next group, Colin's hand was up, and I wanted to give Venkatesh and Daniel this opportunity to say a few words, the sponsors of this project, and then I'll hand, if Colin you still wants to say something, then
yeah, Daniel, go for it.
Yeah, absolutely. Yeah, I think the work that was done was excellent. I think any opportunity to bring modeling into the clinic to give non-modelers the opportunity to work with models, and at least the findings of models, is always something that we should try and push forwards. And I think generally the opportunities in the ADC space for mechanistic modeling are huge. The ability to inform first-in-human dosing, even in discovery, I think is really, really exciting. And I think Nuran very succinctly pointed out that there are quite big gaps in the literature, and anything we can do to try and sort of fill those in, be that through modeling or through actual data capture, would be really exciting. And yeah, well done, guys. It was a great presentation.
Thank you.
Thank you, Daniel.
And Innocent, I'll hold my question for the open Q&A.
Okay, thanks all. Thanks Team Eli-Lilly 1. And Colin, can you please share the slides for the next team? So next is Team Eli-Lilly 2. Rashida, and so please welcome Nada El Hofi, an associate professor of pharmaceutical technology from Egypt, and Rashida Nakamyuka, who's a biomedical scientist from Uganda, and they represented Team Eli-Lilly too. Again, in collaboration with Eli-Lilly, this project focused on natural disease progression modeling in Parkinson's disease, using data from the Parkinson's Progression Markers Initiative for the PPMI study. They applied nonlinear mixed effects modeling techniques to better understand how Parkinson's symptoms evolve over time in non-treated patients. Over to you, Nada and Rashida.
I hope you're seeing my full screen.
Yes, we can see it.
Okay, welcome everyone. And I'm glad me and Rashida to be presenting today the outcomes of the project that we were working with Eli-Lilly. I'm Nada Al-Houfi and my teammate is Rashida Nakamuka. Our project is under the title of Natural Disease Progression Modeling in Parkinson's Disease using a publicly accessible dataset. This was under the mentorship of Innocent from the APT core team and Eva, Gary, and Drew from Eli-Lilly. I will take you through a rapid introduction and overview of the project together with our objectives, our timeline, and then I will hand our methods and results for Rashida to present. Parkinson's disease is a complex neurodegenerative disorder characterized by a number of progressively declining aspects of the motor and non-motor symptoms. And although there are in the market some symptomatic treatments for the disease, yet there is no actual cure for the disease. This necessitates the presence of a natural disease progression model that can guide every clinical decision that could be taken throughout the journey of the Parkinson's disease patient, where it can help to understand the dynamics of the motor and non-motor symptoms that may evolve over time with the untreated patient, as well as predicting the insight and the trajectory of the anticipated decline for newly diagnosed patient. And the most important of which, to help within the research and the clinical trials to identify targets, choose our endpoints, or even present a patient selection criteria specified for each clinical trial. And finally, when there are target molecules to treat this disease, we are ready to evaluate these treatments by establishing a baseline for comparison of this symptomatic and disease-modifying interventions. The data set we are working with is the Parkinson's Progression Markers Initiative, which is an international collaboration. The PPMI main outcome is the Movement Disorder Society-sponsored Unified Parkinson's Disease Rating Scale Score, the MDS-UPDRS. The PPMI dataset consists of 4 parts. Part 1 deals with the non-motor aspects of the daily life, whereas parts 2, 3, and 4 deals with the motor symptoms of the disease. Part 1 itself, it is reported by both the clinician and the patient, where it is covered by 13 questions, 6 of which are answered by the clinician during the diagnosis, and 7 are answered by the patient. Whereas part 2, it is self-reported by the patient himself through answering 13 questions. Part 3, it is reported by the clinician through examination for the motor functions and their disabilities, like the tremors, rigidity, and gait, things like this. And they are 33 questions to be answered by the clinician. And finally, part 4, it represents the motor complications that the patient can face during his journey with the disease. And it is represented by 6 questions that are answered by the clinician. All of them have the same scoring system, ranging from 0 to 4, where 0 represents the normal condition and 4 represents the most severe condition. So the aim of our work is to establish a natural disease progression model through establishing structural models for the natural disease progression, following by examining the effect of the different covariates on the disease progression. We focus on part 1, which is the non-motor aspects, because it is a combination of the clinician and the patient out views. So we modeled part 1A for the clinician, part B for the patient separately, and then we modeled them in a combined form. Our workflow started with a heavy literature review, followed by exploratory data analysis and cleaning of the data sets, because we had like 6 data sets to work on, followed by construction of the base models, their evaluation, choosing the best base model to go on further with, into the covariate evaluation and their effect on the disease progression, and finally into the reporting stage and drafting of our publishable paper. As we can see from the exploratory data analysis, for both the clinician on the left side and the patient data set on the right side, we can see that both of them had 0 inflation. So we had to modify the data set by adding an integer, because this is categorical data set. So we had to add an integer 1 to shift all the scores by 1 in order to be able to perform any sort of log transformation or log analysis or log modeling for this data set. When we studied the missing data and analyzed our data to check where are the missing parts, there were 4 covariates that had almost no missing data sets, which were the sex, race, age, and ethnicity, whereas there were 2 other covariates, which are the sexuality and the PathVar count, which represents the genetics here, that had more than 50% missing data. So we focused on the complete data sets for now, for the sex, race, age, and ethnicity, to study them and their effect on the disease progression, and we are keeping the sexuality and the genetics to a later time after imputation of the missing data. This is a summary of our covariates with their mean, with their different subtypes, stratified with the sex. And as we can see, we have around 3,836 individuals after removing the missings. And then I will hand it to Rashida to present our methods and our results. Thank you.
Thank you, Nada. I am Rashida, and I'm going to present the rest of our presentation. So for structural modeling selection, we tested 3 structural models. We tested the linear progression model, the exponential progression model, and the logistic progression model. For each of these different models, we tested the combined error, the proportional error, the additive error model as well. And then we also tested whether we had correlation within the parameters or not. For selection of the best model, we based on the model with the best goodness of fit plots and the lowest objective function value. We did the model building utilizing monoliths and Linksoft connectors in R programming. We shall begin by showing you the results for the clinician data. And we found that the logistic progression model best described our clinician data, and specifically that was the proportional error model. As you can see, we are comparing between the goodness of fit plots and the visual predict and the VPC of the combined error model and the proportional error model. And there may not be so much of a difference, but when you look at the estimate of the additive error in the combined error model, it is very small. So we decided to stick with the proportional error model. Next slide, please. This is also a comparison between our chosen model and the best model from the exponentials. And you can see from the VPC that our chosen model is a better description for our data. We also went ahead to compare with the best linear progression model. And still, when you look at the VPCs, we had a better VPC in our chosen model. So we went with the logistic progression model with the proportional error to describe the clinician data. We went ahead to carry out covariate analysis utilizing Samba in monoliths. And we found that there were no statistically significant covariates. And so we stick to our
base to our previous model, which is the logistic progression model, as our final model without any added covariates. Going ahead to the patient data, we found that still the logistic progression model, we can move on to the next slide. We still found that the logistic progression model was the best model that described our data. In this case, the combined error model was the best model based on the objective function value in comparison to the proportional error model as well. You can see that the objective function value for the combined error model is lower than that of the proportional error model. And we got a good VPC. We went ahead to do covariate analysis for this model as well, and we utilized Samba. And we found that the age and sex were statistically significant, and they had an effect on the disease status at baseline. Also, when you look at the plots of the parameters versus the covariates, in the first plot, we can see that there is an increase in the disease status at baseline with increasing age. This is a comparison between the VPC of our base model and our final model after adding the statistically significant covariates. And you can see that there is an improvement in the VPC, especially in the upper percentile. Then we went ahead to stratify the VPC for the final model by sex, that is the female and the male. And we can see that the females showed a higher disease burden compared to the males. Then we also combined these two parts, as Nada explained in the first part of the presentation. So we combined the part 1A and part 1B to have a combined part 1. And we also went through the same process of modeling to come up with the best model that describes our data. And we found that the combined error model was the best model that described our data. And in this case, we can see a comparison between the candidate models, that is the proportional error model, and the combined error model. And the combined error model had a lower objective function value. So we went with it as our best model for the combined part 1 data. Then we went ahead to do covariate analysis for the combined part 1 and found that the age and sex also had an effect on the disease status at baseline, which is the S node. And even in the plots of the parameters versus covariates, we can see that there is increasing disease status at baseline with increase in age. This is a comparison between our best model and our final model VPCs. And in the final model, we can see that there is an improvement in the VPC, especially in the upper percentile. We also stratified the VPC of the final model into the females and the males. And if you can notice, we have a slightly higher disease status at baseline in females in comparison to the males. Next slide. In conclusion, we modeled the natural disease progression of Parkinson's disease using the PPMI dataset, and we modeled part 1A and part 1B separately. Then we combined them at the end and found that the females showed a higher disease status at baseline, and the older individuals also had a higher disease status at baseline in comparison to the younger ones. Thank you so much. And in a special way, I would like to thank the APT faculty for supporting us through this very rigorous period. And then I'd like to thank my teammate, Nada, for being very cooperative. And also thank Innocent, our mentor, and our sponsors, and everyone who has contributed to this, the APT. Thank you so much.
Thank you so much all.
Thanks, both. I know Eva has a very urgent meeting. Eva, do you want to say a few words before you go and then come back?
Yes, no, I just wanted to really thank Nada and Rashida. It was a lot of work. I mean, here, obviously, they've caught up very quickly. We were dealing with a very large data set with a lot of questions. So they've done amazing job, and yeah, very proud of them. I will be back in 30 minutes and address any other questions that are coming. Thank you.
Thanks, Eva. So like before, the Q&A is open. If you have any questions, please put them there. And so as we wait for questions, Drew, do you want to comment on the project? Okay, as we await questions, I have a few of my own.
Okay.
So I looked at the population predictions. And I think usually when you're doing models, we know all models are wrong, but some are useful. So these could be the best models for the data set. But do you have any? Do you plan to try other models like for the kind of data you showed, where you have very many zeros? Do you plan to try models like, say, beta regression?
Yes, actually, we're planning to do this, and we did part of this actually, analyzing using the beta regression because to some extent it's a recommended model for this type of categorical data set, and we expect that we will have also good or maybe better models using this model.
Okay, and to add on that, so Ray has asked, any plans to use causal frameworks such as ACM for the future with the kind of data you have?
Sorry?
So any plans to use causal frameworks in the future with the kind of data you have?
Causal
models, causal frameworks such as SEM.
Yes, maybe we will do this, but it will depend also, it will not be only our decision, it will be a collaborative decision between all our mentors. So we have lots of plans and we have lots of extensions for this project to do, but we will be limited with the approved ones.
Can folks hear me online now? I apologize, I was having headset issues.
Can't hear you.
Yes.
Wanted to say that Nada and Rashida did an excellent job on this project. It was, as Eva pointed out, a massive data set that they had the responsibility of combing through. Maybe Nada and Rashida, I know you guys talked a lot about your work modeling the part one of the non-motor experiences of daily living. Maybe, I know you guys at least had a chance to explore maybe part three, if you could look forward and think about, what other modeling you might apply to that data set and what you might expect to see.
Okay.
Yes, definitely.
Yes, we shall do that.
We are planning to track the 3 parts.
Yeah.
Thanks, Drew. Another question. So were the same number of covariates tested for both the patient and clinician data?
Yes.
But we have other covariates that we need to impute the missing parts in them first before starting to using them, and we expect them to have also a significant impact on the disease progression and to make the models look better, even better.
Okay, thank you. Another question. What informed the choice of covariate model, in this case, Samba?
Yes, we use Samba, yeah.
So why Samba?
Because it is, there were 3 options actually in the monoliths, which is the SCM, the Samba, and the Kozak. And all of them, they go for the forward addition and backward deletion. But they differ in what they measure during this process. But actually, because we are dealing with a huge data set, so one of the aspects that we chose based on was the fastest. We used the fastest that gave us reliable results. That's why we used Samba. Rashida, would you like to add something to this?
No, I think that's it. We majorly wanted to explore all the coverage as fast as we could because initially we actually had quite a number. So we decided to use Samba because SCM was taking quite a lot of time given the datasets that we had.
Thank you. If no further questions, I'm going to thank you again for the presentation and hand over to Colin.
Thank you so much.
Thank you.
So thanks, Innocent, and thanks Nada. Thanks to the Eli-Lilly team and the mentors. Some of them need to move on to other meetings, so they'll drop out of the conference call right now. My suggestion is that we go into a tea break now. We're running ahead of time, but I think this would be good anyway, that if possible, we could finish ahead of time. So if we go on to tea break now, so this on my time here, I see it's 6 minutes past the hour. So we'll reconvene at 15 minutes past the hour. So with that, I would ask you to take a rest, stand up, get some blood circulation going, and we'll reconvene at 15 minutes past the hour for the next two presentations. The first of those will be on bioequivalence, and then the next one will be the malaria project. So talk to you guys again soon. So welcome back, everybody. Trust that you had a good break. And let me now hand over to Samir, who will introduce our next team.
Thanks, Colin. I hope you can hear me well. So I have the pleasure to introduce our next team, Team BE. It's a topic that is very important and relevant to the African continent. So the team consists from Natalie Sang. She is currently a researcher in a clinical setting, and Francis Shiumya, where in addition to his academic roles, he managed a pharmacy, and he wears several hats, research hats. So both worked on this project, and we had the opportunity as well to get access to a real BE trial that was run in Africa years ago, based on work that Colin started studying the bioequivalence of several fixed drug combination for the treatment of tuberculosis. So I will leave it with the team to walk you through this project.
Thank you very much, Samir, and good afternoon and good morning, everyone, from wherever you are. We are very excited to present the work that I've been doing in this APT fellowship. And my name is Francis Chumia with my colleague Natalie Sang, and we have been working on the review of open source software and licensed software for bioequivalence assessment, and also looking at the implications for training and technical audiences. The mentors for the project were Samia Mokas, Colin Pillay, and also Ochuko Rere. So just a bit of a background. Bioequivalence is something that is of very importance, particularly in Africa, because this is the setting whereby the majority of the medicines are actually generic formulations. So globally, regulatory agencies would mandate the sponsors to actually do some bioequivalence assessment before a drug is allowed on the market. And as part of the review process, they do reanalyze these data to confirm that the formulations are really bioequivalent. But these steps are not consistently applied, particularly in Africa, because of some of the challenges that the African regulators do face, including limited infrastructure, high cost, lack of skills, etc. Next, please. But the good news is that African regulators are very keen to improve the way they do their bioequivalent assessment, as we have seen that some of the regulators have come up with the guidelines and made them accessible so that the sponsors could be able to follow. But however, sponsors do not really follow all these guidelines, as we can appreciate from the studies that have been done in South Africa, that there were a lot of deficiencies in the submissions that were done in terms of the bioequivalence sections. And having a chat with some of the regulators, it seems that the challenges are similar across the African continent. Next. So the objectives of this APT project was that we could be able to contribute to the increased efficiency in regulatory review of BE data and also the reports, and that we can be able to propose suitable software that can be well used by non-technical scientists, and so that we can also set up some training program for non-technical scientists in Africa. So this is the overview of the workflow. First, we had to select some candidate software for our project. So we selected Phoenix, PKanalytics, and R, and I will present more about this software. But we went ahead to validate the software using the choose dataset. So this is a dataset that was already published, and it has also been used to validate various software. So we thought this was a good platform to follow suit. Then further, we did a non-compartmental analysis and also the bioequivalence assessment, but we applied the quantitative data, and we chose this data set because it was from the Africa, and we thought it was putting things in the right perspective. Next, please. Just to talk about the software that we used in the project. So PKanalytics and Phoenix are the first appearing here, and these ones apply the graphical user interface. They are relatively easy to use. For PKanalytics, it produces very attractive plots. On the other hand, Phoenix is commercial standard for NCA, and it has been widely accepted by a number of regulatory authorities. As compared to R is not commercial as PKanalytics and Phoenix is, so it's absolutely free, fully available, just downloading from the website. But the challenge is that it requires some programming skills because it uses some coding for you to do the data analysis. So we validated the softwares, as I already said. The choose dataset had already applied and was already applied and being used to validate software like Phoenix and R, but it did not include the PKanalytics, which was part of our project. So went further to validate PKanalytics as well. Next, please. Yeah, so we use the core data, as I said, to do the NCA and BE comparisons. And this was a three-way crossover study design with 3 rifampicin formulations. So there was Rifafor, which was a 4 fixed drug combination. There was Rimactazid and also Rimactin, which was used as a reference. To start with, we transformed this data set to become a 2x2 data set. And we had 2 separate data sets. So this is the correlated data analysis, and we can appreciate that really in real-world setting, data would have some peculiarities. And this is what we had. We noted that there were some other participants who did not receive the drug in both the periods, and also there were some detectable concentrations at time 0 for some of the participants. Next. And so we did a head-to-head comparison in terms of the non-compartmental analysis, and then subsequently also did the BE. So this is what we have from the non-compartmental analysis. From our 2 data sets, it yielded that the other data set was balanced and the other one was unbalanced. And this is because for some of the participants, as I already said, they already only received 1 formulation. And also for some of the participants, they had insufficient data points to plot the AUC infinity. But what you should note from this analysis was that when we repeated the same analysis across all the 3 software, the results were exactly the same. So we also did the BE analysis. And the results for the BE analysis, the first one is the balanced data set that we had. And for the balanced data set, when we had to compute the point estimates for the geometric mean ratio for AUC last, Cmax, and AUC infinity, we found that across all the three softwares, the values were exactly the same. For the confidence intervals, we found that the results were the same only to the fourth decimal point. And here we just presented the 4 decimal points just for precision, but for regulatory requirements, only 2 decimal points are enough to present the data. So the results were actually very good across all the software. But for the unbalanced data set, we noted that PK Analytics had different results as compared to Phoenix and R. And this is because PK Analytics was able to kick out the participants who did not actually complete taking the drugs in both the periods. So here is the summary of the comparison after we had an experience with these 3 softwares. So in terms of the cost, Phoenix and PK Analytics, as I already said, that they are commercial software, so there's a cost associated with using this software. Of course, for PK Analytics, it's partly free, but only available for academic use only. On the other hand, R is absolutely free, even for the regulators. They can be able to download the software. In terms of the ease of use by the non-technical users, there is minimal training that is required for the use of Phoenix and PK Analytics, and this is because they are using the graphical user interface. However, for R, you need a bit of an intensive training because of the use of the coding and the likes, and so you need to make sure that people have been well trained for them to flexibly use. There is commercial support for both Phoenix and PK Analytics, but for R, you can have extensive support from platforms like RAN and GitHub. As we saw from the data that we had, the handling of non-completers is supported by Phoenix and R, but for PK Analytics, it was not able to support non-balanced data sets. In terms of the transparency and reproducibility of the data, there is some moderate reproducibility in Phoenix and PK Analytics because of the limitation that some of the steps are using the graphical user interface. But for R, it's actually you achieve high transparency and reproducibility because of the use of the coding. So by the end of the day, you have got a full script that can actually ensure that there is high reproducibility. So from this point, I'll hand over to my colleague to continue with the presentation.
Thank you, Francis. So once we had completed the comparison of the various software, we attempted a model-based bioequivalence assessment using the 3x3 dataset. And this is just because with model-based bioequivalence assessment, you might be able to reduce costs by reducing the number of samples to be taken or the number of participants to be used. And also it may be of benefit to African regulators to have knowledge of the same. So generally you prepare your data and do an exploratory data analysis, after which you develop a population PK model. For us, we chose to use relative bioavailability and added a few covariates. Relative bioavailability is of importance because this is going to be a bioequivalence study. This population PK model is then validated using VPCs, goodness of fit plots, and bootstraps, and then it can be used to simulate various bioequivalence scenarios and estimate various key bioequivalence metrics. And just for clarity, modeling is usually going back while simulation is using the model to go forward and look at various scenarios. So we cleaned up our data to ensure that it would run properly. We then tried various structural models. We tried a one-compartment model, a one-compartment model with a lug, a one-compartment model with a lug and frill, a two-compartment model, a two-compartment model with a lug, and a two-compartment model with a lug and frill. Frill is relative bioavailability. The two-compartment model with relative bioavailability was the best model because it had the lowest objective function values. We then tried various error models. We tried an additive error model, a combined multiplicative error model, and a combined error model. We selected the combined error model again because it had the best objective function values. We then moved on to select covariates. We started with the occasion covariate. And after we added it to the model, we created some box plots to ensure that we had accounted for this occasion variable properly. And as you can see, we had accounted for it well. We then moved to formulation. And when we added the formulation covariate, it accounted well for clearance and relative bioavailability, but not for Ka. So we also added Ka to formulation to Ka, and as well as relative bioavailability and the lug. And we also added the weight covariate to volume and clearance. So when we compared our model's performance to the traditional bioequivalence assessment, we found that it did very well. On the left, you can see the full bioequivalence analysis by traditional means, and on the right, you can see the model-based bioequivalence assessment. And when you look at the geometric mean ratio for formulation 1, the AUC last was about 78.4835 with the traditional bioequivalence methods, and with model-based bioequivalence methods, it was 81.1829%, which was pretty close. For formulation 2, it was 105.685, and for the model-based bioequivalence assessment, it was 103.1589. So the two are pretty close, and this shows that model-based bioequivalence assessment can be a good option for people to use. So when we looked at how well our PK parameters were simulated, we found that AUC last, for example, was augured well with our simulated AUCs. The medians are also close. As you can see on the plots on the right, the black dotted line is the observed median and the blue line is the simulated median, and they were very close. And this trend repeated for Cmax as well. The distribution using the line graph was similar for both the observed and the simulated, and the medians were also close. We also attempted a gamma absorption model and a transit compartment model, both of which did not emerge to be the best models. So currently we have completed our we're almost completing our validation of the models. We are planning to run a bootstrap, after which we can then simulate various virtual bioequivalent scenarios and estimate various metrics.
Scenarios that are going to be simulated. So as already mentioned, a balanced data set with full sampling. The other one will be a balanced data set with full sampling. And we'll also try to see sparse sampling with 2 to 3 points, and also for non-bioequivalent formulations. And so from our experience, we have tried, and we are in the process of developing the training materials. And so this is the course that we are proposing to administer among the African regulators. So the course name is Bioequivalence and Regulatory Evaluation of Generic Medicine. And so the course is designed to equip drug regulatory professionals with practical skills for analysis and evaluation of bioequivalence data for registration of generic medicine. It puts emphasis on the use of open source software, such as R, and the interpretation of the results for regulatory submissions. So this is the design of the mode of delivery. We planned that it could be a blended approach, whereby you can have some online modules and also meet on the face-to-face. So whereby on the online modules, we can have some recorded lectures, discussion forums, and also some reflective assignments. But on the face-to-face, we plan to have some hands-on tutorials, demonstrations, and discussions so that people could be equipped with hands-on practicals on the use of R and how they do interpret the BE data. So this is the proposed curriculum. We are going to have some prerequisite modules which the participants would be allowed to take before we go into the core modules. So some of the prerequisite modules include the clinical pharmacology and the pharmacometrics, so that people should have a basic understanding on this, fundamentals of biostatistics, model-independent analysis, and bioequivalent assessment. And some of these prerequisite modules are already available on the PMXAfrica module platform. So the core modules that we are in the process of developing include the principles and design of bioequivalence studies, global regulatory requirements for BE and legislation for generic medicines, practical bioequivalence, looking at R, and also based on request, depending on the various levels of the regulators, we might also include some of the commercial softwares like Phoenix. And the last module would be on the evaluation and interpretation of bioequivalence data for regulatory decision-making. So in conclusion, we see that a reanalysis of bioequivalence data by African regulators is very essential, as this would actually give us some assurance and to make sure that we have got safe and efficacious medicines on the African continent. The open source software like R, they offer a very, very great opportunity because of reproducibility. And also we have seen that they're able to give us very good results, which are just comparable to what Phoenix and other softwares give. And so therefore, we are assured that what they are analyzing is of regulatory standard. Programming skills may be a barrier for some of the regulators, but that's why now we are coming in to propose that we can have a good training tool and a training program whereby this can actually be a platform where everybody else can be able to access and use R for bioequivalent assessment. So thank you very much. That's the end of our presentation, and I would like to thank our mentors and other APT fellows who made very good contributions to our work, and everybody else. Thank you. We'll be glad to take any questions or comments.
Thanks, Francis. So we'll open it for questions and comments. We'll check if we have some in the Q&A. So yeah, that's an important question. What is the difference between balanced and unbalanced data set, and what's being balanced? So Francis, what your take is?
Yeah, so
what's meant by balanced? Yeah.
Yeah, so in the 2x2 data sets, the participants are able to be subjected to both formulations. And so when we say non-balanced data set is whereby maybe some of the participants were only subjected to the drug in one period, and then there was no data for the other period.
So that can be one definition. If traditionally bioequivalence was being done using, you can even imagine calculators, but balanced, there is two levels in a sense. You might have different numbers of subject in each treatment. That's one level. You still have everyone completing both period, completers, we call it as well. And another level of imbalance is when a subject drop out. Basically, they are only participating in the first period of a 2x2 crossover. So some regulatory agencies require completers only, and then you pull all data with non-completer as a sensitivity analysis. But yeah, that's what we mean by balanced and unbalanced. Checking if there is additional comments or questions. Yeah, as soon as a question pop out. As a starting point, which software would you recommend? I am biased towards R.
So join the club, right? All of us are biased towards R.
Well, it depends. I would say if you are comfortable in coding and you can use R, why not? Like there have been many the team reference at Schul's publication. Basically, they compared and there's several publications out there comparing, say, SAS BE results to R, to Kinetica, etc. So R can be used in a regulatory framework. And
Yeah,
that's
sure.
what the team.
Just to add,
Yeah, go
just
ahead.
to add on what Samia is saying, because we are looking at this in the African context, and the major challenge that African regulators have is lack of resources, and that is why we are also biased towards because being an absolutely free software, accessibility is not a barrier for every regulator, regardless of how much resources they have.
And that
Yeah.
really my attempt at making a joke. So Simba is thank you, Simba, for that comment question. Simba is one of our African brothers who is now I'm not sure what your latest job role is, but Simba has spent time within the FDA. So in a way, it was a good question, maybe a loaded question from your side, Simba. So us as modelers, that's why I say we're biased, but in a way, what Francis and Natalie were looking at was, the ability for a non-technical audience to be able to use some of these things. And in a way, R is difficult for a non-technical audience to use. But Samir, I started to talk just when you were going to say something, so I interrupted you.
Yeah, I want to mention that some commercial software offer you what we call a validation/qualification package. I don't know, maybe Phoenix Wind Nolan has a validation suite. You can buy it separately if you will. Now, if you use R, that doesn't mean you don't have to qualify your installation and validate whatever you are doing. That's on you. You need to document, make sure everything is reproducible, making sure that whatever R is you are using is qualified. It's not something like that anyone can tamper with and modify how ANOVA confidence interval are calculated. So that's important to know. In a regulatory setting, you still need to validate whatever you are doing. And Khalid has a comment with AI. R is more easy. With a grain of salt, you need to know what you are doing. You can easily get wrong code or ask R about how to do bioequivalence. It will spit LME code, but maybe not the correct sequence within subject. You need to double check whatever you are doing. You can't just trust AI blindly. But it is easier to get started for sure. Checking if we have additional comments. Yeah, Colin, do you have comment on this? On the AI can solve it all?
Yes,
mindset.
I'm gonna pop a recent Harvard Business Review article on this topic of work slop. And basically we see a lot of AI generated work. In a way, I'm actually a fan of AI, and I think there's going to be huge opportunities for AI to be used and large language models to be used within, for example, the initial triage period of reducing the workload of a regulator. And then one could take it a little bit further and actually do some of the technical checks. So we've been playing around with it, and I have to say that I'm impressed with stuff that it took us a long time to learn how to do, that AI is able to then digitize plots, extract the data points, and actually then recalculate the area under the curve and assess the Cmaxes. But make sure that there's a human in the loop is my rider at the moment. Thanks, Amir.
Thanks, Colin. So, see if there is any additional comment. No, that's answered. All good. So yeah, if there is no more questions, thanks BE team, Francis and Natalie, and I will head over to the next presentation.
Thank you, Samir. Yes, so it's my pleasure to introduce you to both Ismail and Alan Kengo, both postdoc students, one from Cairo, Egypt, that's Aya, and then Alan from Uganda, Kampala. We've had the fellows had the privilege to work on data that was provided by our sponsor, MMV, so Medicines from Malaria Venture. And essentially the data provided was preclinical data, some in vitro and some from murine data or humanized mice. Sorry, I see the slide as skid mice over there. And they use this data essentially to look at how predictive PK or sequential PD modeling can be at predicting essentially PD parameters used for essentially estimating efficacy or estimation of the dose to be used in human. But without stealing any of their thunder or anything, I'll hand over the floor to our fellows, and they can walk you through the presentation. Again, thanks to the fellows and thanks to our sponsors. Thanks.
I will share the screen. Please confirm if you can see our screen.
Yeah. Thanks, Alan. Thanks, Alan.
We do.
Yeah.
Good
Aya,
morning
please,
or good afternoon, everyone. I'm thrilled to present our final APT project entitled Development of a Preclinical PKPD Model for Pyrrolonidine. This project was conducted by our team, myself, Aya Ismail, and my colleague Alan Kingo, under the supervision of our mentors, Vinan from APT Core Program and Robin from MMV. In this presentation, I will begin by introducing the project background and objectives, followed by an overview of the pharmacokinetic model developed for pyrrolonidine. After that, my colleague Alan will continue with the pharmacodynamic component, explaining how the PKPD models were integrated. Next, please. Malaria is still a major global problem. In 2023, malaria affected more than 200 million people, with 94% coming from Africa. The biggest problem is the majority of the deaths were reported in children less than 5 years. So why we still need new anti-malarial drugs? Although there are marketed anti-malarial drugs, resistance has emerged across all major anti-malarial drug classes. In addition, the current treatment protocol contains at least 2 drugs with multiple doses, which may affect the patient adherence. So developing a new anti-malarial drug will help to simplify the dosing regimen. Moreover, to block malaria transmission, it is imperative to develop new anti-malarial compounds target gametocytes. It is challenging to develop the animal model for malaria, as the parasite infects only human erythrocyte, and the disease process in the mice is different. For instance, in Plasmodium infection, the human spleen plays a key role in cleaning the parasites, a process that doesn't occur in animals. Another obstacle is the logistics and the practical limitations of conducting studies that intentionally infect volunteers to evaluate a drug's safety and efficacy, making such trials difficult to perform. Therefore, translational PK/PD model could serve as a bridge between preclinical data and clinical trials, helping to identify promising compounds before initiating costly trials, and therefore accelerating antimalarial drug development. We shall demonstrate the use of preclinical PK/PD model for antimalarial compound to guide prediction of efficacy in human using byronaridine data. Byronaridine is an old antimalarial drug that is currently used in combination with other antimalarial drugs in a 3-day treatment for malaria. The mechanism of action of byronaridine is to bind to free heme and inhibit hemozyme formation, leading to the accumulation of toxic heme within the parasite. This causes oxidative damage and ultimately kills the parasite. On the right side is the summary PK profile of byronaridine, which shows extensive distribution to the red blood cells and the liver, explaining its effectiveness in treating malaria. Most importantly, that it has a long half-life. That's why it is used in combination with other antimalarials. This slide gives an overview of how SCID mice experiment was conducted. All the data used in our PK/PD model were generated from these mice, SCID mice. SCID mice are immune compromised to allow malaria infection, and the mice were inoculated with human RBCs infected with Plasmodium. And after 3 days of infection development, byronaridine was administered orally in different doses for 1 up to 3 days. The drug concentrations were measured for 7 days, and parasitemia was monitored every 24 hours for up to 60 days. Therefore, our project had 2 main objectives. The first objective is to develop a pharmacokinetic model of byronaridine in SCID mice that describe the drug exposure profiles following different dosing. And the second objective is to drive a PK/PD model linking the byronaridine concentration with parasitemia and to assess its translational value by comparing the model-derived EC50 from our SCID data with the EC50 estimated from human study. This comparison allows us to evaluate how well the preclinical model predict clinical efficacy. Here is the summary of study animals included into our dataset, presenting the number of animals, days of treatment, number of animals showing parasite reduction or regrowth. In total, we included 22 animals in the study, receiving byronaridine doses ranging from 0.5 up to 100 milligram per kg. Most of the animals were given 1 dose, 6 animals were given 2 doses, 3 animals were given 3 doses. Only 3 animals showed no reduction in parasitemia, that's highlighted in red in the table. In contrast, doses above 10 milligram per kg resulted in a marked decrease in parasitemia, while lower doses showed initial reduction followed by regrowth over time. These animals are crucial for EC50 estimation to characterize the concentration-effect relationship within our PK/PD model. So the first step was to build our PK model. From our exploratory data analysis, it has shown that the dataset included animals treated for 1 day, 2 days, and 3 days. The concentration time plots showed that administration of byronaridine for 2 or 3 days resulted in higher plasma concentrations compared to a single dose. We started to build our PK model using Phoenix software, and our final PK model, which provide the best individual fits, was a 2-compartment model with first-order absorption. We added inter-individual variability was incorporated on all PK parameters, allowing for correlation among all the PK parameters, and we used a proportional residual error model to describe the unexplained variability. In this figure, the figure shows the result of individual fats stratified by the response. The first group, or the first plot on the left, showed the no response group that has no reduction in parasitemia. The second plot is for the recrudescence group of animals that showed regrowth after initial reduction in parasitemia. And the third plot was for the cured animals that showed a cure up to 60 days. Most of the animals showed a good individual fat between the observed data presented as markers and the model prediction presented as solid line, confirming that the two-compartment PK model captured the concentration time profiles well. Based on this result, the next step was to fix the PK parameters using sequential PK/PD modeling and to proceed with developing the pharmacodynamic model to describe the pyrrolidine effect on parasitemia over time. At this point, my colleague Alan will present the next section and focusing on the process of building and evaluating the PK/PD model. So over to you, Alan.
Thank you very much, Aya, for presenting the first half of our project. First, I will present the raw data, the raw pharmacodynamic data that we had. And as you can see on this slide, we could easily classify the animals or the data from the animals into 4 groups. The first being those that were not treated and hence showed natural progression of the infection. And then those that were treated but did not show significant reduction in parasitemia. And then third is the group of animals that showed a reduction in parasite growth, some of which got to below the limit of quantification, but then later showed regrowth of the parasites. And then the final group for which the reduction in parasites was permanent, or what my colleague called the group that showed cure, for which even after 60 days, there was no regrowth that was observed. Please note that I cut off the x-axis here for presentation purposes because the rest of the data there is below the limit of quantification. So the first thing I'll do is to try and describe the PD model that we use to describe this data. But before I do that, I would like to reiterate that the method of quantification of the parasites that was used in this study, which was flow cytometry, was such that the observations could not tell apart the living and dead parasites. So the quantity of parasites that we see is both for the living and dead parasites. And as a result, we had to stratify our model into 2 compartments. That's the PD model, one for the living parasites and another for the dead parasites. So for the living parasites, we allowed for growth of the parasites with a rate constant, which we are calling GR in this case, and also a reduction for the animals that showed a reduction in parasites that is driven by what we call kill here, that is basically an Emax function with a maximum effect, which is the Emax. The concentration being driven by the PK model that was explained by my colleague earlier, and an EC50 that was one of the biggest deliverables of this exercise. And then the killed parasites were kind of characterized by this compartment, which I'm calling dead parasites here. And one thing to note here is that in the case of the animal model, like again, I mentioned earlier, the clearance of parasites is not similar to that is observed in human beings, and we had to characterize that by another first order rate constant, which we are calling clearance para. And as such, we had these 2 differential equations. Of note here is that the baseline parasitemia is what we are calling P-live, but maybe I can explain that later with the next slide. But basically, the parasites that we observe, or the live parasites here are referred to as P-live, and then they grow at a growth rate, which we are calling GR here. But then it's limited to a maximum should I say, like parasitemia, beyond which the growth rate kind of slows down. And then, of course, for the first compartment, it's the concentration of parasites is reduced by the kill, which is described by the Emax. And the second compartment we can see here is constituted by the parasites that are eliminated from the first compartment, and then these parasites are cleared by a clearance para that we can see over here. Again, our observation is a combination of both the 2 and we used a proportional error model to explain the random unexplained or the residual error. Yeah, so first, during the model development, we soon realized that not all the data could explain all parts of the equations. And in this case, we picked the data apart and kind of opportunistically used the data to explain different parts of the equation. In this case, using the data that had or that was derived from animals that were not treated, we used part of the first equation to explain or characterize the growth rate and the maximum parasitemia. And as I said earlier, our data was collected, or like as my friend mentioned earlier, our data was collected from 24 hours onward. So the live parasites started at the P base, which is basically the parasitemia 24 hours. This is how that model fit our data. And this is a plot of both the individual predictions and the data, the individual predictions being the lines and the data being, the observed data being the markers. And as you can see, this model described the data pretty well. Our estimations of the baseline parasitemia was about 1.3%, the growth rate of 0.036 per hour, and Pmax of 24.8% per hour. Of note, we had variability in all parameters because the data could support estimation of that variability. But as you'll see later, not all the parts of the or the groups of the data could support these parameters. So to fit the other data, we had to look for good estimations of the other parameters like the Emax here, and we utilized data that was published within our sponsors' portfolio. And in this particular case, they did a parasite reduction ratio experiment where they exposed the parasites to different concentrations of the drugs, including the study drugs, which are here MMV 00025 and times the 100 times the IC50 or 10 times the IC50, and then observed the log reduction or the reduction in the logarithm of the viable parasites over 48 hours to determine a parasite reduction ratio of over 48 hours, and then from that an Emax was estimated. So we use this Emax because as you'll see later, the concentrations we achieve in our data could not estimate the Emax. So we found a lot of help in using previous data, especially for this case, the Emax. The other parameter that we had to look for a good estimate for to get the model working was the EC50. And to achieve this, we plotted the model prediction, the PK model prediction of the data beyond the observed about 72 hours. So we used that model to simulate the concentrations beyond that, and plotted it on the same graph with the observed PD data, which is here. You can see the markers and the black line. And then from the point at which the regrowth happens, which is estimated to be the concentration at which the EC50 is observed, we were able to have an estimation of an EC50 of 7 nanograms per mil in the animals that had regrowth. So this initial estimate again helped us very much in getting the model to work. Next, I'll show the model fit, and then I'll conclude with the estimates. This is how the model explained the fit of the animals for which no treatment was given. And as you can see, it does a fair job. From, again, these are plots of both the observed data, which is the markers, and then the model prediction, which is the line. And on the y-axis, we have the parasitemia, which is a percentage of the red blood cells that have the parasites. And on the x-axis is the time. The next group are the animals for which no marked or significant reduction in parasitemia was observed. Again, these are very similar to the previous group because most of what we observe here is the growth rate. And again, the model predicts fairly well. The next group, which was a bit more challenging, are the animals for which regrowth was observed. And as you can see, our model, after the development that I've tried to explain previously, explains the data pretty well. Again, the markers are the observed data, and the model prediction is the line. And as you can see, we capture the regrowth very well, which was very helpful here in estimating the EC50. And finally, the other group are the animals for which there was no regrowth, that my colleague referred to as the animals that showed cure. And for those, as you can see, most of the data was fit well, except for some of these for which less data was collected in the earlier parts of the for which less data was collected earlier. Okay, so next I'll show you the estimates of our model, and as you can see here, I present the data a bit together. In this case, again, grouped in those 4 groups I talked about. So we had to fix the maximum parasitemia from the value observed in the data, which was from the animals that were not treated. Similarly, the growth rate, because most of the animals do not have the data to support this. So these 2 parameters are fixed. And then the p-base was estimated to be 1.53% for all the data. This is what I tried to show here. And then this is the variability that was observed in the baseline parasitemia. And this is the uncertainty that we observed after a non-parametric after a parametric bootstrap was done. The next estimates here are the Emax, of course, which I said we could not estimate with this data alone because of the concentrations that were used. But again, the Emax was fixed to what was estimated from the parasite reduction ratio. And the EC50, which is the most important parameter that we sought to derive out of this modeling exercise, was estimated to be 21.7 nanograms per mil. And the clearance para, I put them on this plot because they are the ones that drive the kill that we, or the reduction of the total number of parasites that we observed. Again, most importantly, I said the Emax was fixed to what was estimated using the parasite reduction ratio. The EC50 had a lot of variability, and this was the 95% confidence interval. And the clearance para, we found that we could not estimate variability around it significantly, and then we just estimated the typical parameter with a 95% confidence interval. In summary, or in conclusion, we managed to characterize the pharmacokinetics of pyrrolidine from a skid mouse model. And the 2-compartment model was with inter-individual variability and correlation among all the ethers was useful in describing the pharmacokinetics of pyrrolidine. And then our estimate of the EC50 from the pyrrolidine PK sequential PK-PD model was 21.7 nanograms per mil. Later on, when we compared this with the human data that was available from our sponsors, using the translational factor calculated from the plasma binding of the drug in mice and human beings of 0.54, we estimated a human EC50 of 11.7, which is again not so far away from what was estimated using the human data. But the key takeaways from this project are that PK-PD modeling was used to describe exposure-response relationship of pyrrolidine in mice, and this could be an alternative workflow to generate or to predict efficacy in human beings that can be used to propose first in human doses that can then be used to effectively develop antimalarials and yeah, so that's the end of our presentation. I would like to thank my colleague Aya for being available for us to do this work, and Vinant was very helpful, especially with the software, and Robin for guiding our projects, and then our APT mentors that were also available for questions whenever they came up, and colleagues for the moral support and help here and there. And of course, the sponsors that made all this training and project possible. That's the end of my presentation, or our presentation. Over to you, Vainant.
Thank you, Alan. Thank you, It's a great presentation. I really appreciated the work and appreciate the hard work everyone put in. And as you mentioned, great support from our sponsors, MMV, and obviously the data available to use. I see, I'm just going to look through the Q&A. I see there's a couple of comments. Simba said, commented on insightful, basically paper information. And then there's a comment on the translation factors, a fraction of unbound. So maybe you can just elaborate on this, is what the fraction unbound refers to. When we're looking at basically comparison between, say, a mouse and versus the human or human infected data.
Yeah, do you want to give it a shot, and then maybe we can ask our sponsors that do this quite often to, yeah.
Yes, the translational factor is a plasma protein binding factor because pyrrolidine is highly bound to the plasma protein. So this translational factor is provided from our sponsor to translate the estimated EC50 from the SCID mice to the clinical. But we see that maybe there is another factor that can modify more the estimated EC50 from SCID mice. So over to you, Natalie, if you have to add anything
I
about
see Natalie's
this.
hand is raised. Yes, thank you.
Yes, okay.
Yeah, okay, I think it's working now. You can
Yes.
hear me. Yeah.
Thanks,
okay.
Natalie.
Yeah. No, firstly, Arno, thank you for your work. So we've not met, but yeah, I'm Natalie working with Robin, and no, it was a very clear presentation. For the translation factor, it's a little bit more complicated than plasma protein binding. We need to transform because, in fact, take into account the fact that the SCID blood is made of SCID plasma, human red blood cell and mouse red blood cell. So we need actually to then convert it into just the blood EC50 to take into account the blood to plasma ratio, both in mouse and in human, and the plasma protein binding in the two species.
Thank you,
I had
Natalie.
as well a question, if I may.
Absolutely.
Alan, you presented the data on parasitemia, and you in the data exploration, you actually tried to estimate the EC50 from the curves. And I wondered, how did this value, the value you obtained in the data exploration, compare with then the value that you estimated?
Yeah, so as you can see, the value from the data exploration, and I again want to emphasize that the initial estimate was very important in the fitting of this model, because we tried several times without before we got to this, and found that we always had the model struggled to fit, especially the data with regrowth. Yeah, but as you can see, the initial estimate was about 7 nanograms per mil, and our final estimation was, as I said, I think about 3 times this, which was about 21.7 nanograms per mil, considering all the data are modeled together.
Yeah, and you read the concentration at the recrudescence time.
Yes.
Do you want to go for this?
Yeah, we estimated the EC50 from the animals that had recrudescence with plotting with the prediction from the PK. So it was a wide range, by the way, starting from a low dose around 5 nanogram till up to 20 nanogram per mil. It was a wide range, but in average, it was around 7 nanogram per mil.
Yeah. And do you expect the EC50 to be at that time, or is it an approximation?
We estimated it on the inflection point when the regrowth started.
Yeah, so I think to answer the question further, yes, it's an approximation. Yeah, and then maybe the background behind it is that at the EC50, you expect that I think there is 50% efficacy, and then basically the point at which the growth seems to take over rather than the kill. So that spot when that happens is what we basically ended up estimating to be the initial estimate of the EC50.
Yeah, it would not be the concentration at the time when it recrudesces. It's actually what we call the MIC, the minimum inhibitory concentration, which corresponds to having the killing rates equals the growth rates. So it may not necessarily fall on EC50 depending on the properties of your compound, but that's why that it can give you a first estimation. Yeah, no, thank you very much. Yeah.
Thanks, Natalie. I see Robin's also joined the call, so I also invite him if he has any comments. I also see the Safas posed a question. This was more a question based on the Emax and what was fixed. So perhaps Alan and I can speak towards the parasite reduction ratio experiment and maybe elaborate a little bit more on why that was required to give a good estimate on Emax rather than using the murine or mice model data on its own.
Maybe I can start by saying that yes, we were. I mean, we did this with so many iterations and trials, but the EC50, for example, rather the Emax, for example, it might have been that in the mice or in the data that we had, we didn't have high enough concentrations to actually be able to estimate the Emax. So we struggled to estimate that until we got an informed estimate from the in vitro study. As maybe I could have mentioned in the trials that in the introduction, this project was nested within a wider drug development kind of workflow where we had data from the in vitro studies, data from the murine model like we have now, and of course the human data. And the purpose was to compare the results if we use the to see how POP-PK or how the PK/PD modeling could inform that process. Yeah, so we did not have high enough concentrations to estimate the Emax within the data that we had, and we used the, I'll just quickly go back to this slide, the parasite reduction ratio experiment to estimate the Emax. Aya, you wanna maybe hint more about this?
Yes, the Emax was estimated from the in vitro data. After estimation, the IC50 for serial dilution for the bionaridine, and then they used 100 IC50 and 10 IC50 of the bionaridine to estimate the Emax. So it's already derived from the in vitro, not from the in vivo data.
Thank you. Yes, yeah, and perhaps just to elaborate a little bit on that as well, is that obviously the in vitro study here is a specific drug action on the parasite, independently on the parasite, so not via the murine data. So the kill was reflected directly on the parasite. So if we're measuring it obviously in the skid mouse or mouse model, again, as you pointed out, it's really a combination of live and non-viable parasite that we're really measuring. So the in vitro data gives us accurate information directly on the parasite. Hence we can use this data and fix it on the Emax. But thanks, yeah, that's great work. And again, Robin and Natalie, if you want to add anything, yeah, welcome. The floor is also yours. I don't see any further questions at this stage, unless you want to add further comments.
There is one more, Fernand. It's from
I'm-
Wheeler, who's worked on this project already.
My apologies. Let me see. Hi, yeah, here we go. So Mwila asked a question, commented, great work, yes, agree. And then he was asking a question on what was the estimated human dose and how was it related to the volunteer infected studies?
Wheeler, that's a wonderful question, but unfortunately, this is one step a bit further from
Yes.
our intended objectives. Yeah, so our objective here was just to derive this EC50 and then maybe as further work to simulate, eventually to be able to simulate the doses in human based on that, and then I think that would constitute work down the line. Yeah, it's a good question, and it sets the ball rolling for what could come out of what could be done with this kind of work. Back to you, Vainand.
Thank you, Alan. Thanks, Aya. Yes, I don't see now anything further. So unless Natalie and Robin, you have anything further you want to just add on or comment on, feel welcome.
So Vainand, I'm just wondering whether Natalie or Robin could maybe pick up on that last question from Wheeler, given that pyronidine is already in the clinic.
So the estimated human dose for pyronidine that was found in the volunteer infected study.
So I can maybe answer that then, Colin, if you want an answer. So for I think Mwila has a good question, but the question of this would be more, the VIS study isn't necessarily a study designed to find, or to directly find an efficacious dose, but it's a study to inform a PK/PD model, which will then be used to predict an efficacious dose in patients. And so the kind of way the translation workflow is put together is that from the SCID experiments, it's an EC50 which is estimated, and it's that EC50 which is then used to design the VIS experiment. So what's more interesting to compare is the EC50 estimated from the SCID versus the EC50 estimated from the VIS study. And yeah, I think that's the last little point which needs to be done in this project. But we had a look with the fellows already. We had a bit of a sneak peek, and it looks like it's pretty close already. I think it's within twofold or less.
Right. Thank you, Robin. Okay.
Yeah, I would have slightly different answer. In fact, I still think that we effectively predict the dose from the mouse PK/PD model, but in this specific case for pyronidine, we did the work a little bit the opposite way. Is that, as you said, pyronidine is already on the market, and therefore it was not so much in our portfolio. Normally, we apply this to new compounds, and this one was done the reverse way, if you want. So pyronidine was developed before even MMV existed, and so without all these studies and all this modeling in place. But then we needed it because we considered the pyronidine to combine with new drugs, and therefore we needed to have this PK/PD model. And then it was so we did actually the VIS after the clinical trial, and eventually we did the mice PK/PD modeling, because then we thought it's only a small effort to have one more compound and check whether the translation is fine or not. So that's something that is going to be very useful for us. And yeah, after, if there is either if Aya and Alan have time to calculate the human dose, or if someone else wants to volunteer, we are interested.
Thank you, Natalie. Thanks very. Okay, I see there. If Colin, if we have time, I see there's another question posed by Safa. She was asking, do you think developing a PBK or physiologically based PK model will help translate this data and the PK/PD model to predict model in human? By this way, it may help to include different criteria, for example, of the plasma protein bindings.
Yes. I think this is a good idea, and I think it's already done in an MMV Solar software platform. They use the PPK and the physiochemical properties of the compound to predict the clinical. And I think integrating the PPK and also the PK-PD model will give us a good estimate for the prediction and the efficacy in clinical. So it's a good idea, yes.
Thanks, Aya. Thank you. And I'm going to see if I can steal more time. I see Simba's raised another. He says this is especially important for back translations, which aims to improve uncertainty in the first human dose projection. I would say that's more of a comment based, but yes, I think that's exactly what Natalie was commenting on just now. So it's more of a case of retrospective and looking at something that was on the market already and basically having information now available for future combinations, especially with a partner drug like Pharmacometrics. Okay. I don't see anything in the chat at the moment or the questions. Yep. So that will be it from the MMV side, and unless Natalie and Robin anything extra to add, but if not, then I think we can move on. Yeah. Thank you very much again. Thanks to the fellows and thanks to our sponsors. Thank you so much.
That's great. We're into an open discussion now. So Robin, you can stay, Natalie, stay, and anybody else that wants to join. Like, let's see if we can stretch the limits of Zoom. So this is an open discussion. We've got 30 minutes, but we don't need to use the time. And here it's really anything goes. So you could ask some questions about any of the presentations that you've seen already. You can comment, you can whatever. So those of the colleagues that are here as attendees, if you like, if you raise your hand, then we should be able to allow you to talk as well.
Yeah, Colin?
Yes, go for it.
Yeah, I don't know. I believe one of the interesting questions that Paulo has raised is about integer-bound models. So I don't know if the team I know you tried many models, including this more advanced integer-bound and boundary. So, Paulo, were you satisfied by the answer, or did you need additional details? If Paulo can speak. Did we unmute Paulo?
I've unmuted him. I've put him onto the panel as well. But
Okay.
maybe we might be putting him under pressure, you know. He might be doing the usual of multitasking.
No, that's fine. But no, yes, that was an interesting exercise. We really tried to look at different possibilities and how to properly analyze this type of clinical score data. But yeah, if Paulo has no additional comment, maybe we can move. So maybe I will add a comment on the bioequivalence type of project we presented. As the team showed, it's very important to have accessible software that student can use, that regulatory agency can use, and that's usable by non-programmers. That was one of the goals. Leaguesoft has made PK Analytics available to us. They've been providing free access to software in various settings. One difficulty we face is we couldn't, like the BE module is a little bit, let us say, not flexible, meaning you can't bring in your PK parameters already pre-computed. You can tell it to analyze using a general linear mix effect model like we can do in Phoenix BE module or in R. But yeah, it's good to have options. And so that's comment. And what's also interesting, Pharmacometrics Africa has been running a regulatory training course. Maybe Colin can add more. So we had first-hand access to regulators across the African continent, listening to what kind of challenges they face, how they are collecting bioequivalence data, how do they look at it, the challenge of having to deal with possibly fake trials, fake data. So I thought that that's interesting as well. Colin, any comment on that?
No, I can. I noticed that Natalie's thing lit up, but if anybody wants to talk, just maybe raise your hand since there's so many of us here and we can open. So Samir, I'm pleased that you actually drew attention to the bioequivalence. So I'm going to say the usual things that I say about it. You know, we as modelers consider this stuff to be boring because it's, you know, it's sham analysis, slope, height, area, moments, but people focus on the fact that it's sham. It's not real stuff. You know, we want to draw models. So I was really excited that this team, Natalie and Francis, just took that to the next level in terms of their approach to the project, their interest in it. And so they presented their work last Thursday to this class of about 35 or 40 regulators from across Africa. I'll pop into the chat in a minute a link to the training course that we did. And we were pleasantly surprised, but reassured actually that there's not a lot of trainings like this available. So in fact, it surprised me. I thought there was a lot. If we are mistaken, can you please, to all the attendees, pop into the chat any training course that you're aware of that exists? Because I'm a big believer that we should not develop something if it exists already. So yes, Samir, that was great work there.
Sorry, yeah, I just wanted to comment or pose it also to the BE group regarding one of the objectives were basically to create a learning module or learning platform or teaching platform to the regulators. But I was just putting in or thinking about it in context of as far as my understanding and work of the regulators, their time is exceptionally limited. So was there any thought on how to make this training material, training course, format more flexible or you know, online or recording, or was there any consultation with them to see, you know, how would they best, you know, like the training done? Because I mean, I think this is absolutely needed and required and useful, but it's just how do you deliver? So was there any feedback from that meeting regarding this? Thanks.
Yes, of course. The regulators also expressed similar concerns, and what it opened our ears is that we need to keep on engaging them and do a deeper and thorough analysis in terms of the needs assessment and how they feel about it, so that it can be more convenient to them as well. Yeah, so those are the things that we'll be looking forward to.
So just to add on to that, one of the things that we were trying to look at is how they do their work and what their workflow looks like, so that we can target our interventions to specific places, so that we're not just throwing things and hoping that they stick as well.
Thanks, Natalie.
And then I'll just add one more, you know, so most of you guys know that the FDA, the US FDA actually reanalyzes data across the board almost, but particularly with bioequivalence. But the European Medicines Agency doesn't do that. So in a way, it's also important to try and figure out, you know, even though we would like it to be done, that to see whether this really is something that is both needed and will be need to be done in terms of reanalysis of data within the African regulatory agencies.
Just to add a colon, I mean, you mentioned about the bioequivalence, but recent example from Lilly for the EMA NDA submission, they do ask for all the files, modeling files, whether they have capacity to reproduce or not, we don't know, but we have to share as a sponsor to EMA, same package what we shared with FDA as well. So that's happening in the EMA.
That's fairly new. But
yeah,
so that's a new development.
Sure.
So they are planning to wrap up capacity and maybe do some pinpoint analysis. Previously, Europe asked us to do any kind of analysis. They just ask us, do this, do that. They didn't do it themselves, but now they will have this capacity. Muila has his hand up. Muila?
Give me a second while I unmute Muila. Muila, you should be able to unmute yourself now and talk.
Okay, yes. Are you able to get me?
Yes, we can hear you, Muila.
Okay, yes. Sorry, I had to step out for another meeting. So, but I just have maybe a question, maybe a general comment from all of you on the presentation that was made by Rasida and the other person, I've forgotten the name. Yes, but they were presenting on the Parkinsonism. Yeah, so, and the data they were using, it was like sort of like scoring, like where you score, and then you use that data like to model. And I could see that she tried about 3 models, and yeah, there was a little bit of misfit and all that. But my general question is, how do you take into account like human error? Because that sort of scoring is not more like you are measuring the machine, like concentration. Concentration, I mean, you can't be biased. I mean, it's a machine that is measuring, but I'm talking about scoring whereby maybe a clinician looks at this and then makes a score maybe of 2 or 3. So in modeling, how do you take care of that? The fact that there's human error, there's too much human error involved here, compared to a biomarker that can be quantified maybe by a machine. Like how should the approach be?
Love the question. I don't know, Venki, whether you would want to help in any way.
Again, from not Parkinson's context, but I did my PhD in schizophrenia, where we use the rating scales, PAN score. We had total subscales and things like that. We had the similar issue, actually. For example, we identified 2 regions. One, I think the Eastern European versus Western European. Eastern European tend to had the baseline slightly higher than what we had from the Western countries. There was kind of a bit of an inflation there. The way we handle, because only that region was slightly off in terms of the baseline PAN score, where they're eligible to get into the trial only because the baseline was above 90 rating scale. So the way we did was kind of use that as a covariate and see what is the magnitude of the effect was different or not between the rating scales from these 2 regions. That is the way we kind of accounted. We know we couldn't explain why it was like systematic bias or not, but we accounted as a covariate effect there. I think, yeah, it is subjective, as you said. One way, I think another one is a residual variability where unexplained variability can be explained. That is a way we handled. I think it could be the same with Parkinson's as well. Again, that's my kind of recollection based on my PhD work.
Okay.
Yeah, and I posted a link to a family of models we call item response theory models. So, and that can happen in many applications, namely in, say you want to assess, I don't know, intelligence or anything that you cannot measure with a machine, as Muila mentioned. So the family of this model has items where maybe questions can be like you ask a patient for how he's feeling, or a clinician is assessing the patient. But bottom line, you need to analyze the data in a probabilistic way because they are prone to error. And maybe when you do survey, you realize, oh, I've been asked the same question in multiple times in a different way. And that's on purpose, because you want to ask it in a different way to remove some of the error. So, but yeah, for more information, you can look at this IRT's model. There is several statistical packages that can do that. You can code this in non-mem Monolix or Phoenix. But yeah, these are the type of models that can help you with this type of data.
Innocent, yours is a comment on the same question.
Yes, just to add on what Samir said. So yeah, so measuring something multiple times. So if you look at that, it has multiple parts. The same part one has sub parts. So if you measure it in slightly different ways and you arrive at the same answer, someone is progressing, then at least it points you to what to. So if you use various sources of data. So what this team did, they first started with part one. Of course, they looked at it separately, and then they combined it. But later, they also want to combine the other parts. Now, how you combine also matters, because if the way they combine the first part one, they are showing 50% weights. But later you can give different weights depending on how accurate or how good you fit the model. You can realize that, for instance, clinician data or patient-reported data is fitting the model data probably, and of course you have to discuss with the people who are clinicians or experts at that. Instead of giving it 50% weight, you could say maybe we believe this data is more realistic, so let's give it, for instance, 80% weight, and then give the rest 20 weight. But that is what. Something which is planned, and it's not a small project. But as I was mentioning this, I thought about real world data, and generally when you're working with real world data, measurement error is inevitable. But that's precisely what tests whether a drug really works outside control settings. And it's also why sometimes you don't really need to rely on data alone. That's when sometimes you have to have semi-mechanistic models which try to incorporate physiology so that you don't rely on the data, because the data will always have some noise in it. Thank you.
So I'll just add one last comment on that. And so I think most of the answers that you've had have come at the analysis stage. And I think you could also, or you should, address the topic at the design stage. So in other words, collect data that looks at the same outcome, but from different perspectives. So it's really nice. Innocent used the word real world data. I think Samir, you used the word patient. And I think more and more we're collecting data from the patients directly, as opposed to a clinician's assessment, makes a lot of sense.
Yeah, we call this patient-reported outcomes. Like back in the days, we didn't take this seriously, what I would call it. But more and more, yeah, there is a separate type of endpoint that can be used in clinical trials to approve drugs, and some of which are patient-reported outcomes. So, and there is an FDA page on it, so we can double check it. And Simba had an interesting comment, and I replied, basically with his experience at the FDA, he was saying, anything that will go to the label, FDA will do the analysis. They will double check. You are recommending a dose for renal impairment patient in your label, FDA will double check it and will push back. If your dose is too high, it's too risky for this patient, FDA would not accept whatever you are proposing into your label. So there'll be some kind of negotiation going on.
Yeah,
Simba?
Yeah.
Yes, I hope you can hear me very well.
Yes, we can hear you, Simba.
Unfortunately, I don't know what's happening with the video anyway. It will be good to be seen as well. So that's exactly what happens. So the process is, it starts, okay.
And in a second or two, you'll be able to see and hear him.
Yeah, the drug labeling negotiation is very, very important for the sponsor and the FDA as well. Yeah, again, it's patients first, right?
Right, absolutely. So
Yes.
here's what happens. So the whole process does not only start and end at submission level, let's say pivotal studies. It also boils down to all your earlier phases. So when you submit this data to, let's say, end of phase 2 to FDA, they would also want to see all your first human doses, especially for the PK and PD selection of biomarkers. And when that analysis is done, that is very important. Why? Because in the labeling language, there are certain sections where, in most cases, the sponsors, they would also want to include as many indications as possible. So that way, it won't be based on the available data. And at times, for example, when you have this bone marrow depletion, that is a cause of concern in certain drugs, and the sponsors, they don't include that. So when we modeled the data when I was at FDA, we actually include all these adverse events based on concentrations, to then inform the sponsor that you should put this one as a precaution or this one as a contraindication, depending on the indication of the drug and also the target population. It could include lactating mothers, it could include pregnant women, or it could include adolescents. Mostly your first application is in adults anyway, 18 years and above. And rarely would people consider geriatrics above 65, but in some cases, we could use simulations based on the data available at NHANES. So there will be much more robust analysis of the data that you submit. Not only that, they just take your model, see the results, and then give you the feedback. They also do a lot of analysis around it to inform the sponsor and the applicant as well.
So thanks, Simba, and good to see you, although at the moment you've got a very bright background, so we can't
Right,
see
so
how,
let me
whether
find
you still
some.
have the youthful looks that we know we sent you from Africa to the US with.
Yeah, I'm trying to find a better seating position right here.
There you go, there you go. That's great. Thanks, Simba. And as Simba starts to, that's absolutely great. I'm wondering whether anybody has any additional themes that you'd like to discuss. We've got about 13 minutes of normal time left. Anything that you heard within the presentations that somebody wants to start up as a theme or a conversation? And as you can see, colleagues that are there as attendees.
Yeah, I had the comments last question. I'm not a specialist in QSP. I know the team mining QSP models from literature had some struggles extracting the information. So I'm not sure if there is some standards on how to share knowledge and data and models, especially this more complex, many ODE models. Like extracting equation from a paper can be daunting and long and prone to error. So I don't know if Venka or anyone working more in QSP, like what is the current state of affairs? Are people sharing codes or QSP models? Is there a preferred language? I'm just curious to know how can we ease the knowledge transfer. You publish your paper, you have all your equation parameters. Why don't you share the code to enable people to enhance the model and keep going? So
I mean,
maybe some comments, yeah.
Yeah, so I think maybe Innocent also can chime in here. So Brett's paper for the QSP of the ADC was the kind of like what we wanted to go with first. But since our APT fellows wanted to have kind of familiarity with the modeling, we started with the Shaw model with the 4 equations. And certainly, I think they were able to reproduce everything, but the units were slightly challenging what units they used in the in vitro assays and things like that. But reproducing the differential equations was straightforward, I believe, but just converting the units was a bit challenging we had. I think nowadays, most of the journals like CPT, JPKPD, they're asking for the codes without any like model code. They're not accepting. As a reviewer, that's the first thing I ask. If I don't see any non-mem codes, the author should provide that, and we are insisting that most of the time. Hopefully that's progressing as well. I think I agree with you. It is very complex, especially when you have like 100 differential equations. I remember back in AstraZeneca time, where we wanted to establish one kind of oncology QSP model with more than 50 or 60 differential equations. And another example was the vaccine model, actually, more than 100 equations. It is a bit of a struggle, but I think it takes time, and also we need to have some experienced mathematician in the background. So I would call it myself as a clinical pharmacologist, but with some mathematics background. But yes, I will get lost. But if you have someone with a good mathematics background combined with you, things will get better. So that's kind of my experience. Anything to add, Innocent, based on the work we did for the ADC?
Yeah, thanks for that. And yeah, so I'm not aware of any standards for what that there's a publication which tries to recommend how you should publish your protocol for QSP models. But regarding reporting, I have I am not aware of any publication. And then regarding the journals asking for model code, many journals do, but I don't think reviewers actually try to run the code. So if you don't run the code, if there is no, I don't think it depends, but so it will be few reviewers. That's what I think. Few reviewers, in my experience, will actually do the actual running of the code, because sometimes you need to have specialist software, and so if you don't actually run the code, you might not be able to find a problem. And sometimes even the problems, because sometimes the authors of the code, they find problems after several years later when they run the code themselves. So sometimes even issues with the code might not be as obvious. So like for the example, the base paper, so all the equations, the initial estimates were published in the supplement, but because they used QSP notebook software, which is from Applied Biomath. So I've never used it. I don't know how it would look like. If it was R, then that would be straightforward too, because and I think that's why it's advisable to most people, if people could go towards R, because with R code is straightforward. You provide the script with everything. And now if someone is using specialist software, you don't really know how they use the equations. The best is to try to understand the equations as much as possible and then try to reproduce them. And I believe this will be done by these fellows. We also have to remember that the project is about 4 months. 4 months is not a very long time. Some of these QSP models, like Mekatesh has said, there's hundreds of equations, and you really need time to understand equations before you go to. So you really need to have time to really understand what's going on. So they were able to replicate a simpler model with fewer equations, and I believe that was very good enough for the given the time frame they had. But hopefully, now that they have finished this part of the project, they will continue with the more complex BETS QSP model, and I believe they'll be able to replicate it.
I think
Thanks, Innocent.
Nuran has a hand up, so I think maybe Nuran wants to respond.
Just small comment. I was hoping for Innocent to say it will be a requirement for everyone to provide a shiny like he did in his paper, so people can click a button and simulate from your model. But yeah.
Yeah, that would be great. But again, you can't force people to use R. So I've seen some code is in Python, and I've been forced to learn Python because I want to use their code. So because hopefully R will take over, but before that, I know even Python has its own interactive but most people will still use commercial software where you can't really know how they are implementing their equation.
Nuran?
Yes, thank you, Innocent and Samir, and Venkatesh. As one of the fellows and working, one of the team members working in this project, I just wanted to tune in also regarding our experience. So yes, it's not just sharing the codes. I think translating ODEs into the different syntax codes may not be the big issue, as probably limits the replication of these models, but it's basically a lot of information that is not well reported in terms of scaling factors, whether they standardized it to volume of the media, volume of the cells. There are a lot of unknown information, and I think that these also have to be reported as well in order to enable future researchers when they're trying to replicate these models to be able to successfely replicate them. So it's I think it's not just a matter of the coding, because as Innocent mentioned, the VETS paper, they did not use R, but I'm assuming that the unknowns or the uncertainties that we had, it's more related to units and scaling factors and so on. So that was my input.
Thanks very much, Nuran. So we've got about 5 minutes of normal time left. Is there any?
Yeah, I see Amira's hand up.
Sorry, I didn't see that. Amira,
Just a quick comment. Yes, it's for Robin and Natalie, because this year the MMV project was a great success and the fellows learned tremendously from this kind of exercise. So are there similar projects in your pipeline that we can benefit from next year? Thank you.
Amira, that's a great sort of theme. And let's, as soon as Natalie and Robin have answered, let's throw that open to anybody that might have projects that might be interesting. Natalie, Robin?
Yeah, we'll try to find another or related topic maybe to see the translation then to the VIS would be a possibility. Yeah.
It's great to hear. Thanks, Natalie.
Thanks.
And Venki, as a sponsor?
Certainly. I mean, definitely we will have this. We want to keep this momentum. We keep getting some kind of questions where we might not have time. I think certainly next year we will have a couple of projects as well. And also internally, we have circulated some of the slide deck which you shared from the APT fellows. They are aware of it. So that's why I think one example was we were able to hire APT fellow into our core group with the US group, which is just show this testament of what APT fellows are doing. Certainly, I think I'm keen supporter of APT fellowship, and I'm kind of asking other colleagues also with Lily to come up with some projects. And I do see sometimes the time is a limitation, like 3 months, 4 months. Yeah, that is a environment we are in, but certainly this QSP modeling, again, how we are using within Lily, we didn't have that kind of R Shiny app. So when we had last year, APT fellowship, which we published, and that Shiny app really immensely helped us for the progressing in some of the ADC projects. And then this year, APT fellows Nuran and Ibrahim also implemented for the in vitro side. That will be kind of built on a little bit once we have some internal resource. I think there's a lot of positives to take forward. And again, I'm very proud of what APT fellows are doing here. Certainly next year, I will have a couple of projects, if not definitely from Lilly group to something different therapeutic areas. Something we have not had, cardiovascular maybe. I know we had this year Parkinson's, and then oncology, something to look into cardiovascular as well.
That's really fantastic. Music to our ears. Does anybody else? Sorry, Vainant, you want to say something?
No, I just commenting, just thanking. But it's always good to have the support.
Yeah, and Simba, I saw your note earlier on in the chat that you want to get involved. This is one way to get involved. To support the fellows, to supervise the fellows, to propose projects. And I always refer to these as the data set that you have in the bottom drawer because a project was killed within your company, but you really have an interest in this, and this is one way to really drive it forward. Simba, you want to respond?
Yeah, sure. Absolutely. I mean, there are so many projects that are also related to that. When you have a molecule that probably gets killed, and you still have this passion to share the knowledge that you probably it could also impact the next molecule as well. But knowledge sharing is also important, and how the models were developed. That will help the fellows as well to understand and also to venture into different kind of therapeutic areas, not only, let's say, infectious diseases, which are most common in our setting, African setting, but also other communicable diseases which are coming up, especially in the Southern African region.
So that's fantastic. And we're right on the top of the hour right now. So I'm going to bring this meeting to a close. And I want to really thank everybody, all of the core team members who are all on screen right now, the fellows in particular. Guys, you did us proud. So you really deserve to have a good break tonight, a good drink of your favorite beverage. This has been really, really fantastic, and we look forward to working together again with people like all of the sponsors that have just proposed these amazing projects that have high impact. Really, they have high impact. So with that, let me bring this meeting to a close. Take care, everybody. Bye-bye.
Bye.
Thanks, everyone. Thank you. Congrats. All right.
Thank you.